Assalamualaikum warahmatullahi wabarakatuh. My name is Wan Nur Aina Farhana binti Wazaidi, S50744. So today I would like to present a review on jellyfish-based novel probiotic products. My supervisor is Dr. Lee Abdul Razak, and I am Bachelor of AGROTECHNOLOGY Science in Aquaculture, Faculty of Fisheries and Food Science. So in this slide, there are contents of introduction, literature review, result and discussion, and conclusion. For the introduction, my background of the study is in the aquaculture production. Disease is a major problem in the fish farming industry due to the Banerjee and Ray 2017. Disease causes serious economic losses to finfish aquaculture in around the world. And probiotic is the best way to treat disease infection due to its positive residue that does not harm the environment. Nowadays, successful jellyfish fishing is carried out in at least 19 countries, with a large number of companies engaged in the manufacture and import-exports of edible jellyfish. So that jellyfish species from the class Scyphon zone is selected to review some potential probiotic in the aquaculture. For my problem statement, one of the major concerns to aquaculture production is the losses due to disease outbreak due to Brishra SS et al. 2017. The alternative to antimicrobial in disease control could be the use of probiotic bacteria as microbial control agents. Public health problem through the bacterial resistance when we use antimicrobials in aquaculture. My significance of study is my study may be a novel to overview about probiotic bacteria from jellyfish species as a potential against disease. They are also a source for determination of the presence of beneficial bacterial probiotics that give many benefits in the aquaculture industry, such as use for disease control. For my objective, to review, summarize, and evaluate current knowledge about probiotic from jellyfish species based on the previous research and for the further application in aquaculture production. For the literature review, I choose Scyphon zone jellyfish because, according to the Cost et al. 2019, the bacterial community composition presents in different parts of the body fish with its umbrella surface, oral arms, and gastric cavity. Blanchett et al. 2014 also state that directly stimulating bacterial growth and potentially influencing bacterial community composition. For the probiotic is a live microorganism that confers several beneficial effects to the host. They also can enhance immunity, help in digestion, protect from pathogen, improve water quality, promote growth and reproduction. Probiotics also have received particular attention due to the high abundance, low cost, and convenience in application due to Hassanifah et al. 2018. Commercial preparation probiotic product. Commercial products are available in liquid or powder presentation, and various technologies have been developed for improvement. One of the first evolution of commercial product focused on a bacterial preparation called BioStart that is derived from bacillus isolates. They are also optimized the fermentation condition to increase the viability and functionality of probiotics, improving performance. Product must provide a health benefit to the host, such as have the ability to survive storage condition and after that in the digestive tract of aquatic species, remaining viable and stable, and finally improving production. For the safety consideration of probiotic products, historically, food-related lactobacilli and before the bacteria have been considered healthy. This conclusion is supported by their occurrence as natural commonsense of mammalian flora and their proven safety in a wide variety of food and supplement products worldwide. However, Patricia et al. 2012 state that probiotic may theoretically be responsible for 4 types of side effects, such as systematic infection, deleterious metabolic activities, excessive immune stimulation, and gene transfer. For my methodology, I review from the previous research. Firstly, I assess the strategy in Google Scholar, ScienceDirect, ResearchGate, and Spingerlin. They are stated that first step is a sample collection and processing due to the cost from M et al. 2019 and isolation of bacterial species, identification of probiotic, characterization of probiotics, and lastly data collection from previous research related to my study. For the isolation of bacterial species, firstly we dissect the body parts of jellyfish species, then we homogenize the sample with phosphate buffer saline, and then we plate the sample onto MRSM media for lactic acid bacteria selection, and then we put the agar into the incubator for 30 degrees Celsius for 3 until 5 days until we see the bacteria single colony, and lastly we maintain the probiotic isolate on TSB supplemented with 1.5% natron chloride. So this is a resection of jellyfish species due to the Leon et al. 2019, which is the X umbrella surface, oral arm, and gastrovascular cavity. Next, this is a quadrimetric strip pattern due to the Sanders at 2012, and this is a Maracus shape and TSB transit soil bar. For the identification of probiotic, we use gram staining to determine cell morphology, colony morphology, and size. We also use using VTech kit for microbiome identification. For the characterization of probiotic, firstly, we use a colony forming unit count CFU, and then we use catalyst test, hydrophobicity test due to Wang and Han 2007, and then adherence to polystyrene surface, kinetics of bacterial growth, pH tolerance, biosalt tolerance, auto-aggregation assay in vitro, and lastly, co-aggregation assay in vitro. For screening of antigenic activity, firstly, we use double layer method, and then we use diffusion method where diffusion agar assay, and then hemolysis activity. For the result and discussion, due to the study of Koskrama et al. 2019, this state that in X umbrella and oral arm shared bacterial groups from alpha protobacteria and gamma protobacteria. Gastral cavity was dominated by beta protobacteria, followed by gamma protobacteria and actinobacteria. This sample shows that the most highly dominant potential probiotic found are gamma protobacteria, especially Vibrio and Pseudoatromonas in all body parts. However, there is not too much result due to its new project purpose. Further investigation of probiotic in jellyfish species is required to understand the relevance of the associated bacteria, specifically probiotic in aquaculture industry. Conclusion. In conclusion, jellyfish species have ability in base novel probiotic products. There are some potential probiotic found in the selected species, and they can use as further application in aquaculture production. So this is my references. And lastly, thank you for your attention.